with regards to annotation, there are different levels at which you can annotate genomes, and CACAO specifically focuses in on this level of annotation where we're talking about the gene products and what they're doing individually as well as in the context of a cell and together. And so we use the gene ontology to do this. It's kind of divided into three things we call aspects, which talk about function, process, and cellular component or location. And the way that the gene ontology is organized is it has, I guess, three key features. It has the controlled vocabulary, which includes a human readable ID, and then the ID is readable by computers, and the name that's human readable with its definition, so you know what is supposed to be used for that number, and then all of these are connected to each other by relationships. So in this example here, you have this Go ID that refers to the term plasma membrane, which has a definition, and it's related to all of these other terms in the tree, in this ancestor chart that we're showing. So the plasma membrane is a membrane, which is a cell part, which is a cellular component. And so for each of these three aspects, if you kind of define them, molecular function is referring specifically to activities or what the protein is doing, and this is shown by data, different kinds of data, and each of them will have their own term, and so there's some more broad terms and some more specific terms. So this is hexokinase activity, very specific, versus just kinase activity. And then for biological process, this is a series of events. It's more than just biochemical pathways, so it could also be referring to something like cell division. It could be referring to transcription, and it can also be referring to things that occur at the cellular level. Cellular component, so I always think of this as location, but location with kind of an expanded definition. So here, like an organelle, mitochondrion, or in a bacteria, the peptidoglycan cell wall, but it also can refer to a complex of proteins, so being in that complex. The ribosome is definitely a big complex. To gather all of these things together, so you put together an entry that's in this very specific format where you have your Go term that talks about whether it's the function, location, or process that it's involved in, what protein specifically you're talking about, and then where the evidence came from, so that's the paper, and what kind of experiment was done, and you put all of those things together to make this Go annotation. So we told you at the end of the last class that you have to pull together these 4 things, and they translate into an ID for the paper, an accession for the protein, and then an ID or code for each of those other parts. So you guys should have the Google Doc with your table that has the information that you put into your table as much as you could last week, or you'll fill the rest of it in this week, and it's going to have your blank annotation tables that you would copy and then kind of the example. So we're going to go through each of these pieces. They kind of fit together. The 3 pieces for the UniProt accession, the pieces for the paper, for the Go ID, and for the evidence code. And these are the 3 websites that you would use. So we're going to go through each of them individually. So for Pubmed ID, you go to a Pubmed page and you bring up the page for the paper that you're looking at. And so it's going to look something like this under the search bar. It'll have the reference, the title, the authors, the abstract, and down here at the bottom, there's going to be the Pubmed ID. So if you blow this up, it looks like this. There's a PMID, and that's the number that you want. There's sometimes a Pubmed Central ID or PMC ID. This PMC number is not what you want. If you try to use that, it will not work, or someone will be able to will have to correct that annotation later. So that's the number that you're looking for your PMID. So often, if you just copy the title of the paper and paste that into Google, it'll take the first link will be to the Pubmed page, and you can get this easily that way. So that one's the easy one. Now if we look at the protein, so this UniProt website, so how many of you tried looking at the UniProt site? Okay, So UniProt it's a big resource for all sorts of things about all the protein sequences that are shared by everyone in the world. And in UniProt, they have this collection of protein IDs. So every unique protein will get its own ID, and that's how everybody can keep track of which one they're talking about. And that'll link it to an organism and all sorts of information about that protein. So if you looked in the paper to find some keywords that would give you a way to search in UniProt, you might have found a couple of different things. There was a paragraph that gave you the names of the proteins. I've highlighted both of these things in red in the CAMI doc as of today. So one of the paragraphs said, ORF37 of Klebsiella phage K5-2 and K5-4 and ORF38. There was also a paragraph in there that talked about, it was at the very, very end of the paper, right before the references. It said nucleotide accession or something like that. And it said, these genomes were deposited in NCBI under this number, and so you might have tried searching that number in UniProt. So usually I'll search something like accession and see if there's any accessions that they give, because that makes it easy. If not, then I'll search the organism or the name of the protein in UniProt. So in this case, if you searched either Klebsiella phage K5-2 or ORF37 or tail fiber protein, you might have tried either way. So you would have searched for kind of this name, and you would want to get to this ID. If the paper that you are reading doesn't give you any information about the specific protein they're talking about, you just need to pick another paper, because it's not going to work for this class. So this is a website. If you've already searched, you can just search in the bar at the top. I want to give you some information about the typical output. So there's two very important things. There's blue and this light blue stuff, and then the gold arrows. So UniProt can kind of be divided into two sections. There's the unreviewed section, which is the light blue stuff, and then there's the reviewed section, or SwissProt, which is the gold stuff. So light blue means there's a protein sequence out there and it's here in our database. Gold means there's a protein sequence out there and somebody has looked at it and they've quality controlled and added some information, and so this is kind of like a good record to refer to and look at these. So there's gold and blue, that's reviewed and unreviewed, and the gold ones are actually looked at by professional biocurators. That's why they get this nice star. Then the other thing is this entry here, this is going to be the UniProt accession ID that you want to copy, and then you might have to look either here or here or here to figure out exactly which protein you're looking for, but you always want to make sure that it's the right organism, because sometimes there are several organisms that are very similar, or proteins that are very similar, and you could get them mixed up if you're not paying careful attention there. So if you want to go directly to a specific page, and you click on the entry ID, and then it gives you something that looks like this. So again, up here, this is the number that you want to copy over into your table, and then double check the organism, and you'll want to copy this name. Usually there's an actual name, but sometimes there's not, because the paper that you're reading is actually where they study it and they find out what it does and they give it a real name that's not just uncharacterized protein. The page will usually have, if there's been some Go annotations added, they'll actually be on there, and so sometimes that gives you a good clue for what you might want to use to look for as a term for this protein, because someone else has used some other assay to show a similar activity, and you might be able to use that as a clue. And then sometimes you'll find synonyms of your protein name. So sometimes if this name doesn't match, the name down here will match to the one that you're looking for. But the most important things that you have to copy to your table are going to be this ID and then the organism. So this, we want to tell you about one thing related to different organisms. So sometimes UniProt will take out of its large collection of organisms that are out there that it has in the database, it will say, okay, we've got a whole bunch of these, but we're going to pick one that's going to be the reference that we want people to annotate to. And so those are called reference proteomes, And you'll notice that this box here is now red. Instead of searching in UniProtKB, where it was blue, it's now red. And so if you're searching for a particular kind of organism, in this case, so see here, if we were looking for this PLED protein, and the paper used a specific strain, but they're making a claim about all Pleiades and all Caulibacters do this thing, you'd want to be like, well, should I annotate this strain, or should I annotate this strain, or what if it's not either of those strains? What do I annotate to? So you look for the reference proteome. So you'd search for Caulibacter and find out which one actually matches, it's the right species, and then you can annotate to the protein in the reference proteome. If you're, anytime you have issues finding a protein, we will help you find the protein. So you can definitely ask questions about that later. So that's for finding the protein. So were you guys able to find any UniProt accessions for your protein when you were searching? No, yes, no. What I guess I would ask you what it is, but I don't have it in front of me. So do you guys have the number in front of you?
No, not immediately.
No,
I
okay.
didn't find that accession number at the bottom. I didn't think to put that in.
Oh, that's good.
Yeah, I put it down.
You found them. What does it start with?
For the K54 438 that I did, it starts with A0A219 YHF3.
Confirmed. That's right. Okay, great. So we'll come back to that, but good job there, Jason. And don't worry, you guys will find them later. So the next one is for your Go term and ID. So this is going to be the word or short phrase that you use to describe either what it does, what process it's involved in, or where it's located. And there are 3 different places that you can search for Go terms. We gave you the link to QuickGO, which is kind of the preferred one, I'd say, that we use the most. But there are 2 other options. There's this Amigo, and actually you can search on the course Go Nuts website. And the goal will be to look for the term and then also get the ID. So the term mainly so that you know what it is, and then the ID is what you're actually going to be entering. If you go to QuickGO, this is another one of those websites where you just search in the box. I mean, that's really the main thing on the first page that you get to. And you can use keywords that come from the figure legend or from the discussion of the results. Sometimes the abstract or even the title of the paper give you the search terms that you'll be using to look in QuickGO. And you will see when you start typing something here, it will kind of pop up some suggested terms. And so you might see immediately that one of these kind of matches what you want, or you might want to see all of the results and go from there. So
right, sometimes the number of possible results is huge.
Yeah, yeah. A few things to point out here. So this is the Go ID, and then we mentioned last week that there's a 1-letter code for each of the aspects of the ontology. So C for cellular component, P for biological process, and F for molecular function. And here it gives you the term, and then this is telling you how many annotations have already been made to that term. So sometimes that kind of helps you see if you're not sure if this is the right way to use this term, you might want to check those, or if you're interested in learning more about something that's annotated to that term, you can check those things.
Yeah, actually, before you leave this, there's a bunch of them right here where that pink arrow, the second pink arrow is, it's saying obsolete. That's because the Go people sometimes decide that a certain term doesn't make sense, so they obsolete it. That means you shouldn't use it.
Mm-hmm.
So it's pretty easy to see if it says obsolete at the beginning.
Okay, so if you click on one of those Go IDs, then it will take you to the page for that term. And there's a really nice section on the side that tells you what is on that page. It's like the page outline. And there are 3 pieces of the page that I'd say you will use a lot. And then maybe this occurring term section. Notice that some of these are grayed out, so not every page has all of these pieces. The important thing that you'll check after you've kind of decided if you might want to check into this term is that you got to look first at the definition to make sure that it fits with the conclusions that are being made in the paper. So, for example, say you're looking at a protein and they're claiming that it's a gram-negative bacteria and they're claiming that it's in the outer membrane, and you found a term that says membrane, so you're like, aha, this might be my term. But it might or it might not. It might be that you want to go more specific because they're claiming that it's an outer membrane protein, not just a membrane protein. So just because the definition fits, that's good, that's a place to start, but then you'll want to go check further down under this child terms section. So in this case, say we're like, oh, tubulin complex, that's fixed, but maybe there's a more specific term. Turns out this has grayed out, so there's no child terms. There's nothing below it in the chart, so this might be the best term. So for synonyms, I find that this is very helpful. So underneath the definition, down here, synonyms, maybe the thing that I'm looking for is this term, and it came up because it was searching the synonyms. Even though this term isn't exactly the same, you'll check that to see if they're very similar. Now over here, I'm showing, this is the ancestor chart for this term, so it's very long, and all of these things upstream would be considered true, but we've arrived at this most specific and most true, maybe the best term. All right, so this is what it would look like for child terms. If there were child terms for a Go term that you're looking at, you would say, okay, so maybe is one of these more specific terms actually best, and if not, then you would stick with the one that you're doing or that you're looking at. In this case, co-occurring terms. Sometimes what happens is you're looking at a paper and it's making several claims, and if you look at this co-occurring terms list, it might give you ideas of what else you can annotate in that paper. And then last thing here is taxon constraints. So this happens where, for example, this term is only allowed to be used on cellular organisms. So if you're reading a paper about a virus, then this term cannot be used. And so in that case, either you can't annotate or you might need to request a new term. So we keep mentioning that like the ontology is changing, they're obsoleting terms get added. We can request new terms, and that also counts as a 5-point, basically the same as if you were to make an annotation. All you do is request a reasonable term. So that's something to keep in mind as well. Sometimes just because there isn't a term doesn't mean you can't get anything out of it. So talk to us about options there. So this I mentioned before. It also lists here a link to other things that have been annotated with any given term. So if you were to click on that link, it might take you to a list that would look like this, and you can see all of the other proteins here that are annotated. You can see that they're all annotated to the same term, and you can see that they come from different kinds of evidence. So in this case, we've got 2 different papers, and then another kind of evidence that's been used that isn't a paper. So that's an interesting thing, right? So that's saying that you could potentially annotate the same protein with one term, but from 3 different kinds of evidence. So just because, like the paper we're reading, say this paper claims that they're tail fiber proteins, and so we find a term and that works and we annotate it. And then another paper like 2 years later does the same thing. Shucks, can't do it. No, you can do it because it's another piece of evidence, and it's just kind of building on the evidence to say that this protein really does what we say. So that's one thing commonly people will say, all right, this is an interesting protein, so I'm just going to find all the papers about this protein and annotate the same term but with 5 different papers.
Can you go back one slide real quick before we
Yeah.
So one extra thing that you can do to help find child terms is there's a chart options box right here. This is another way to view it. You can click this, and then there's a little checkbox that you can click to show ancestors or show children, and then basically it'll populate all the children to a term that you look at. So it's another way to view it on top of just scrolling
That's
down.
True. All right, okay, so that's the Go ID. Now last is the evidence code. So actually we use the same website for the evidence code, which is nice. Just to give you an idea of how this ontology is organized. So we've been talking all about Go, the gene ontology, which has 3 aspects, and that's where we focus. So now eco is another way of kind of standardizing the way that we capture the evidence that was used to make the claim about the protein. And that's why it's called the evidence and conclusion ontology. And it has two branches that come together. And first is the kind of evidence that was used. And that means either, so similarity evidence, meaning kind of like a bioinformatic type thing, author's statement, somebody said so, or experimental evidence. In this class, we're only using experimental evidence to make claims, or to annotate claims. And then the other branch is the method that the assertion was made by. So
Actually, do let them use similarity.
I guess that's true.
We don't focus on it as much though.
Yeah, that's true. We do let you use this kind of evidence, but I sort of classify that under experimental, but it's not. It's like computer experiment versus bench experiment. Yeah. So under assertion, the difference is automatic means a computer was kind of reading through and made a temporary claim about this protein, and manual assertion means that humans looked at it and determined that this was the evidence or conclusion that was made. So for us, we're not computers, so we will always be using terms that fall under the manual assertion section of the ontology. And these are governed in a similar way to Go, so they have very specific IDs and term names and definitions. Those are related to each other by specific relationships in this tree. And when you are searching for eco terms, usually these come out of either the materials and methods section or the figure legend. Sometimes the figure legend doesn't really tell you what they did or how they did it, so then you have to go to the materials and methods. All right, so if you were to click on the very top level term evidence and then look under experimental evidence, you can see that there's a whole bunch of different kinds of categories of experimental evidence. And the best way to search for what you're looking for is to find some keywords in the materials and methods to kind of narrow down where in the tree. Sometimes you want to scan down the tree, but usually searching is a quicker way to narrow it down. If you search in QuickGO, it will look very similar to the Go term pages where you've got the ID, the term, make sure it's manual assertion, has a definition. It'll give you all those other pieces, although the pieces there are less of them. And as we said before, so in this paper, we've got this microscopy evidence here. They're making a conclusion about this particular protein, and we found in the materials and methods that they used stacked confocal images of this particular depth, and it's immunofluorescence microscopy. And so this would be the eco term that we would use. We might have searched immunofluorescence, and we also had to specify that it was confocal microscopy and manual assertion. So that's how you would narrow that down there. Yep.
If they perform like in the same paper report multiple like different experiments on the same thing, can that all be loaded onto the same annotation, or like
Yeah.
different kinds of evidence?
Mm-hmm. It depends. It definitely depends on the paper, I'd say. So sometimes they do several experiments that confirm, and there's really one clincher experiment, and you would annotate to that. And sometimes there's more than one, and you may be able to get additional annotations. And that's actually something that so we only recently started using the eco ontology, and so we haven't actually sorted all of those things out, whether, if they show it in 5 different ways in one paper, do we want to capture all of those things? We might actually want to capture all of them.
So each annotation can only list like one evidence.
Yep, so
Yes.
one evidence code, one Go term, one protein, one paper. Yeah. All right, so here's an example of an ancestor chart for that particular eco term. Now, there's a few special cases for evidence codes where you'll see when you're putting in your annotations that there's this thing called the with from field. And this is something that you might need to use, and if you need to use it,
You're probably gonna want to talk to us.
yeah, you will want to talk to us. And I think it should tell them that they need to fill in the with from field.
It will,
Hopefully.
the with from field will appear in the form when they need it,
Okay. So,
assuming they refresh.
okay. So, for example, say you're doing, the evidence is that you've got some sort of bioinformatic comparison, the sequences are similar, and so in that case, the authors are making their claim that this protein does this based on similarity to this other protein. So you need to capture that in the annotation because it's very dependent on this other protein similarity. So in that case, you would need to put that into your with from field. Sometimes if you're doing, again, another kind of computational analysis, this with from field would refer to the specific kind of tool that was being used. And then the other one that you might need to use would be evidence from genetic interaction. And there's kind of an example of that, pseudo example of that in the paper that we read, where maybe you've got two different mutations in the same strain. And so they're saying that when you've got these two things, then this thing happens. And in that case, it depends on there being two proteins involved, and that's when you would kind of need to have this with from. So it's all going to be case dependent. If you need to do that, make sure you talk to us. All right, so this is the part where we have to tell you that there's a lot of really great science published in many different papers out there in the world. However, not all of those great science papers are good for annotation. And that's because it has to be about a protein. So if there's this really awesome paper that you're super excited about that's like this super nice, great compound that's going to be good for treating cancer, unless it's a protein, it's not going to work. So it's got to be about a protein. Also, sometimes what will happen is people they'll be doing this study and they'll say, okay, when we treat these cells with this compound, then all this stuff is true. And in that case, it's because they treated the cells with this compound. It's not because that's necessarily what would happen. So we generally don't allow you to use Go terms that require it to be in response to some treatment. Also, sometimes there are really interesting interactions that require proteins to be binding to each other, and that's another thing that oftentimes is misunderstood because the Go ontology is kind of confusing about protein binding terms, so we usually don't allow that either. And then the big bummer for some people is that in this class we only annotate normal protein function. So if you're really interested in, the tau protein, or like the proteins for Alzheimer's, they aggregate, they do bad things. Those are very interesting, great science, but not good for Go because we want to know what the protein does normally. That's not what its evolved function is, so that's not what we're trying to annotate. So the other thing is, maybe someone has done this really great study on evolving a protein to be better at making hydrogen gas so that we can have more biofuels to not have to burn so much coal and oil. That's fine, but we can't use that for annotation either because that's not the normal protein. It's an evolved protein.